Good morning, everybody, and thank you for the organizers for inviting me, and Tanya especially for squeezing out a time slot for my presentation in the last minute. So I'm going to talk briefly about setting up a 3D model from a Cliftra patient, mainly for drug screening purposes. So actually our journey, or rather said, the journey of the group in the Clifstra ward started a few years ago when my colleague has identified a point mutation in a panel of ASD patients, and actually turned out that this point mutation is causing the nonsense-mediated decay of the transcript variant 1 of the EHMT1 gene, which will ultimately translate to lower protein amount and finally in basically half amount of methylated histone in the neuronal progenitor cells. Other papers that our group has published has been showing that the basic neuronal markers are equally represented in these two cell lines. But the difference is more in the functionality. When we treat with acetylcholine the cells, we have a higher calcium signal. There is no significant difference with the glutamate treatment. And actually, the present research will concern more the development and characterization of the 3D model of this cell line. So the workflow is containing the NPC cells from both cell lines control and the Clifstra. We plated those ones on a 96 well plate and cultured them for up to 2 months. And we did some characterization using qPCR, immunohistochemistry, flow cytometry, and drug screening. So what you can see here and instantly is very visible that we could assess the 3D from both cell lines, but for some reason the Clifstra cell line is not growing. We were even wondering if these cells in the organoids are alive or not. The genetics was confirmed with the lower methylated histone status with immunohistochemistry, so that was fine. And to check if these structures are alive or not, we just place them on laminin-coated surfaces at day 21, and we could see nice, abundant neurite outgrowth stained for tubulin and astrocyte marker, the GFAP. The difference that we noticed was in the amount of the astrocytes that migrated out from the spheroids. And later on, at the end of the culture, after 2 months, again, we see an abundant, voluptuous neurite outgrowth from Clifstra. So there is no deficiency in this respect. And yes, the organoids are alive. For some reason, they don't grow. So to dissect a bit more this issue, I went back to the NPC stage. And we assess the confluency, and already here you can see with the green line is the Clifstra NPC confluency represented. So we measured this over 3 days using the InQSight device. So the confluency amount is basically significantly lower in the Clifstra versus the control. And we checked with flow cytometry the Ki67, which is the classical marker for proliferation. We see a slight change, like 10% in the Clifstra versus the control. And further, we measured the main machinery responsible for cell proliferation, growth, and protein synthesis, which is the mTOR, phospho-AKT-mTOR pathway. And here again, we see a slight drop in the double positive population. Phospho-AKT, phospho-mTOR is like 15% lower represented in the Clifstra versus the control. And it's the same result in the 3D, only that the differences are much stronger. So basically, we barely have Ki67 positive cells in the Clifstra spheroids, And also the phospho-AKT and phospho-mTOR is underrepresented in the mutant cells. So this might be behind the slow or growth arrest in the 3D organoids that I showed you before. We did a brief characterization using qPCR and immunohistochemistry of these spheroids. What we could see that from the three main cell types, astrocytes are significantly overrepresented. And if we check the neuronal subtypes, we can see that the green one, Chet and the serotonin receptors are higher in the organoids, while the TH is barely present in the Kleefstra. Our results regarding the green 1 expression are nicely correlating with the findings of Frega et al. And this data was presented yesterday in one of the talks. So it's really good that between different cell lines, some of the patterns are conserved and show the similar mechanism. With the ICC, these are really brand new, fresh data. I just got them a few days ago from my colleague. What you can see here that the MAP2 is nicely stained and it's similar in the two cell lines. The outstriking difference is again in the NMDAR. So this is the protein encoded by the green 1 gene. And probably with the quantification of the stainings, we will get the same result that it's also in protein level, it's highly expressed in the Kleefstra cell line. We confirm the presence of synapses in these organoids with synaptophysin. So in both cell lines, we have a nice staining for synaptophysin protein together with the tubulin. So actually, so far it was the characterization of this 3D, and the second part will concern the drug screening and the sensitivity to various toxicants. We used 4 different compounds for this reason. The doxorubicin, which is used in cancer treatment, and 3 other compounds, the paraquat, bardoxolone, and rotenone, which are mainly oxidative stress inducers. And as you can see, the doxorubicin, the bardoxolone, and rotenone did not make a difference in the two cell lines. The difference occurs when we treat the cells with paraquat. The dose response curve shows that the Kleefstra cells are much more sensitive towards paraquat than the control. And I went back to the MPC cells again to see if the difference is the same there. And what you can see here, I plotted the IC50 values. But you can see that in the case of doxorubicin, CDDO, which is the abbreviation for the bardoxolone, and rotenone, the sensitivity of the NPC cells is much higher. So the NPCs are represented with the white bars, and the 3D is represented with the red bars. So basically, because the NPCs are actively proliferating cells, they are more sensitive to most of the toxicants, except for the paraquat, where the trend is changed. So for the paraquat, the 3D structures are much more sensitive than the NPCs, and I don't know why. But obviously, differentiated tissues have lower capacity of oxidative stress response and scavenging these ROSs from the system out. And the other difference, what you can see in the lower table, that while in the wild-type cells, this change in the sensitivity between the MPCs and the 3D structures is 5 times. In the Kleefstra cells, it's like a 15 times fold change between the sensitivity. Basically, at the end of the treatment, we did not have any cells in the dish to measure the ATP. So what is behind this? We checked the genes. Actually, the same results were published a few years back by Riani et al. They found that the mutant flies are sensitive to paraquat, but actually in our case, the gene expression is completely opposite to their findings. We don't have upregulations in the superoxidase catalase GPX genes, not even in the LDH. So basically, we don't have stress response with these genes. And furthermore, the basal level of the LDH, which is a very important gene in scavenging out reactive oxygens, is half in the Kleefstra. And actually, this gene expression fits nicely into the puzzle. As maybe you remember, I mentioned that phospho-AKT and mTOR are downregulated in the Kleefstra, and actually the LDH is a direct target of the mTOR pathway. So probably this is the reason why we have only half amount of this protein. So to conclude, I think it's very important when we try to screen drugs for any disease, it's very important to know what is that drug designed for and what is the best cell type to screen on. Because as you could see here, we have a different sensitivity of the NPCs and a different sensitivity of the 3D, regardless of the genetics of the cell line. So to sum up, we could set up this 3D model, and it's feasible for drug testing. We could also identify differences in the neuronal subtypes in these organoids, and we identified some altered growth and metabolic pathways in the mutant cells. And what we would like to do in the future is to assess the contribution of astrocytes and microglia in the disease, also in 3D model. So thank you very much for your attention.